Good morning, everyone. Thank you very much for all coming here today. My name is Hannah Wardill and I'm a postdoctoral researcher alongside Patrick Hughes at the South Australian Health and Medical Research Institute in Adelaide. And as my title suggests, I'm going to be talking about the mechanisms underpinning barrier dysfunction and also pain sensitivity in irritable bowel syndrome. So I've got nothing to declare. Now, I don't need to tell this audience what irritable bowel syndrome is, but what I will quickly remind you of is that despite the symptoms of altered bowel habit and also pain, we do know that there's no overt inflammation or pathology associated with IBS. Despite this lack of overt pathology, though, we do know that the luminal environment of the gastrointestinal tract is chronically altered with evidence of microbial dysbioses, protease activity, and also low-grade inflammation. And like any condition that's characterised by changes in the luminal environment, in particular microbial dysbioses, there is always the question of the chicken or the egg, which one comes first? Are these factors actively participating in the generation of symptoms, or are they purely reflective of the underlying pathology that's occurring within the gastrointestinal tract? And so this was really the question that we set out to ask, and we decided to model the luminal environment by creating or preparing fecal supernatants, which are exactly like what they sound. They are stool samples that we've collected from diarrhoea predominant IBS patients and also some age and sex matched healthy controls. They are homogenised with a Ringer's solution. They're then centrifuged and the supernatant is collected. And so these supernatants are very reflective of the luminal environment. However, they are without all the gunk that a stool sample comes with. And we were really interested to firstly look at whether these fecal supernatants could actually cause any detectable changes in intestinal barrier function. And we looked at intestinal barrier function using Using chambers, which I'm sure you're all aware of, but they're two independent chambers which are separated by your tissue of interest, and in our case it was segments of the distal colon from healthy mice. And we put the fecal supernatants that we'd collected from our IBS-D patients and also our healthy control samples into the apical chamber of these Using chambers, and we looked at the changes in transepithelial resistance in real time for 2 hours. So this graph here shows the change in transepithelial resistance just with the Ringer's solution in the Using chamber baths, and we have a natural degradation that occurs in transepithelial resistance, and that's completely expected within this time frame within an Using chamber. We then added in the healthy control fecal supernatant, and we can see that although we have that little blip at the beginning, we do see a very comparable time course in terms of the changes in transepithelial resistance. However, when we add the IBS-D fecal supernatant in, we can see that there's a significant reduction in the transepithelial resistance, and this is most maximal at that 2-hour time point. We also saw that when we took the tissue out of the Using chamber, we collected the RNA, we looked at a number of tight junction proteins, and we can see that we've got decreases in a couple, including ZO1, JAM-A, and occludin. And more importantly, we also saw that there was no evidence of any apoptosis occurring in the tissue, which we showed with caspase 3 staining. And this was really important for us to dissect out because we wanted to make sure that the effect that we were causing with this fecal supernatant was specific and sensitive for the barrier and not just degrading the entire tissue. So next we were interested in the mechanisms that might be underpinning this barrier dysfunction that was caused by the IBS-D fecal supernatant. And probably the best characterised in the setting of IBS is the action of proteases. So there are many subtypes of proteases, and they act through a number of different protease activator receptors, or PARs. And so we first did a fairly broad spectrum proteolytic assay to look at the protease activity of our healthy control and our IBS-D fecal supernatants. And we can see that in our IBS-D fecal supernatants, there was significantly increased proteolytic activity in these supernatants. So we then took this through to our Using chamber experiments. This is the same graph that I showed you before with a significant decrease in transepithelial resistance. And we then added in a broad spectrum protease inhibitor cocktail. And we can see that there does seem to be partial rescue of this effect, although this was not statistically significant. And we can see that there's still a portion of that barrier dysfunction that's still remaining. This also was supported by our PCR analysis of a number of PAR receptors or PAR subtypes with no significant decreases in any of the receptors. So we decided to change our approach to it a little bit, and we decided to run an exploratory multiplex assay to look at the cytokine profile of this tissue, of the fecal supernatant, And our multiplex showed an increase in IL-6 expression or concentration in the IBS-D fecal supernatant samples. And so we got a little bit excited by this, and we popped the IL-6 into the Using chamber, and it indeed did cause a change in transepithelial resistance, which was fairly comparable to that induced by the IBS-D supernatants. And this really came as no surprise, because there's a fair bit of literature out there showing that IL-6 is able to disrupt tight junctions and barrier integrity. However, while this was all going on, we also did a confirmation ELISA for the IL-6 concentration in our individual patient samples, not the pooled fecal supernatants. And unfortunately, it showed no statistically significant differences once we'd introduced the higher power of having the individual samples analysed. So this was a bit of a roadblock for us, and unfortunately that's all we've got to at this point with our analyses. But I think we need to probably take a step back and have a bit of a better look at the composition of the fecal supernatants before we move any more forward. So changing track slightly now, another thing that we decided to look at with the IBS fecal supernatants was looking at their effect on nociceptive high threshold sensory afferent nerves of the colon. With the idea that if they're able to disrupt the intestinal barrier, what are they then doing to the underlying nerves that relay pain sensitivity from the colon to the central nervous system? And I have to admit, this is a lot of Pat's work, so hopefully I do it justice, but he's got a really good setup back at SAMRI where we're able to isolate particular nerves from the colon based on their response to different mechanical stimuli. And as I mentioned, we're interested in those high threshold sensory afferent nerves, and they respond to high threshold probing. So once we've isolated these nerves, we can then expose them to the fecal supernatants, and we can look at their electrophysiological properties. So this is an example of one of the representative traces from one of the experiments. We can see here that we do the initial von Frey mechanical probing. We then expose it for 5 minutes to the IBS-D or the healthy controlled fecal supernatant, and we then repeat that probing at the end. Now, one thing that we did notice with these experiments was that upon addition of the IBS-D fecal supernatant, we had an immediate response in that nerve directly firing. And although this is only preliminary data at this stage, we've seen it in 100% of our studies at the moment for the IBS-D fecal supernatant, and we haven't seen it at all in response to the healthy controlled fecal supernatant. And so this suggests that something within that supernatant of the IBS-D patients is actually directly activating these nerves. In addition to this, we also look at the response to that mechanical stimulus, and so we look at the magnitude of the response pre and post incubation with the IBS-D fecal supernatant. And once again, this is only preliminary data, but it does suggest that these nerves become sensitized to mechanical stimulus after they've been exposed to the IBS-D fecal supernatant. So in conclusion, at the moment we're able to identify and conclude that mediators present within the fecal environment of patients with IBS-D are not only able to disrupt the colonic epithelial barrier function, but they're also able to sensitize and directly activate high threshold sensory afferent nerves within the colon. I think that we need to look into the actions of proteases in a little bit more detail, because there is a lot of literature out there suggesting that they're involved in barrier regulation. So I think we need to look at more selective PAR agonists and antagonists to dissect that in a bit more of a sensitive manner. And as I mentioned before, I think we need to take a step back and characterise the composition of these fecal supernatants before we take any more steps forward. However, I think we can show that this data does suggest that there is a luminally driven causative mechanism of barrier function or dysfunction and activation of colo-rectal sensory afferents, and hopefully this will take us a step closer to identifying what comes first, the chicken or the egg. So I'd like to finish up there and thank everyone that has been involved in this study, in particular Patrick Hughes and members of the GI Neuroimmune Interactions Lab back at SAMRI, as well as people from the Royal Adelaide Hospital, as CSIRO, Flinders Centre for Innovation in Cancer, and obviously all the people that donated samples. Thank you very much.